This is Office Hours at Duke University. Today's conversation is with David Goldstein. Professor Goldstein is the director of the Center for Human Genome Variation here at Duke's Institute for Genome Sciences and Policy. His research involves genetics, medicine, and ethnicity. The New York Times has called him a leading young population geneticist who does not shy away from unpopular positions or research. His most recent study, published in the journal Nature, helps explain why some people respond better than others to treatments for hepatitis C. He is not a medical doctor and will not be giving health advice today. This year at Duke, Professor Goldstein will be giving guest lectures both to incoming medical students and an undergraduate course. Viewers can join this Duke Office Hours conversation in three ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. Professor Goldstein, we're here at your office hours. You have this new study out in Nature. It identifies a single gene that has a strong association with whether or not a person responds well to hepatitis C treatments. Let's start with the basics. What is the name of this gene?
The gene is IL-28B, and it encodes an interferon protein, which is an important signaling protein that marshals different kinds of immune responses to control infections.
Very good. So let's summarize this study. People with the favorable version of this IL-28B gene have a higher success rate with this hepatitis C treatment than people with the unfavorable version. It's 80% versus about 30%. That's a pretty big difference.
That's right. It's really quite a remarkable difference for a single genetic change. The background to the study really starts from the observations that are very well known that individuals respond not only very differently to treatment in that some individuals will be completely cured of infection following a course of treatment, and others will not. The virus will come right back after infection. But also with the observation that's been long known that individuals of European ancestry are much more likely to be cured or have the virus eliminated by treatment than individuals that have substantially African ancestry, for example, African Americans. And that has puzzled people for a long time, and many geneticists have long thought that one possible explanation for that is that there is a genetic difference responsible that is itself different amongst different ethnic and racial groups. The other ingredient in considering response to treatment for hepatitis C is that not only does it not always work, but the course of treatment itself is arduous and long. And for all of those reasons, there has long been a strong interest in doing a good genetic study to try to see if one could find underlying genetic explanations for these patterns of response to treatment. And so we and many others have been interested in that, and we finally had a very good opportunity to carry out such a study when the drug company Schering-Plough that markets one of the treatments for hepatitis C approached us about carrying out a genetic study using a trial that they had recently completed. It was not designed for genetics, but it was well suited to carrying out genetics. This trial compared different formulations of interferon alpha treatment for hepatitis C infection, and they had DNA available on about 1,600 individuals from that trial. And for a subset of those 1,600 individuals, they had available good information about who responded well, that is, who was cured by treatment and who wasn't. And we carried out a study that is now relatively standard, which is a Genomewide Association study, where you look at hundreds of thousands of sites in the genome that are known to vary from one person to the next, and you interrogate all of those sites simultaneously in order to get a picture of all the common genetic variation in the genomes of the individuals that you're studying. And we then related all of that genetic variation to who responded to treatment and who didn't. And out of the about 600,000 sites in the genome that were looked at, one jumped up as very clearly having a very strong impact on who responded to treatment.
Very good. And now, I know you're not a medical doctor, but this question is out there. It doesn't take a lot of imagination to think about. You've got this finding that indicates a specific gene is associated with favorable or not favorable response to hepatitis C treatment, that this could have a clinical impact. So what would those steps be to take a finding like this and bring it into the clinic? What would have to happen?
So I think the first thing that I'd like to say is that the way that we think about the diagnostic value of this genetic difference is really that it is something that both treating physicians and patients are likely to want to be aware of as they consider their options. We do not think that there will be a strict algorithm such that you have this particular genetic makeup, then always this course of treatment is preferred. We don't think it'll work like that, but we do think that it is sufficiently strong information that individuals are likely to want to know about it as they consider their treatment options. And I'd just like to sort of draw out some possible scenarios where the IL-28B status of an individual, whether they have the good response form or the poor response form, where it might be relevant. One example is, let's say, a patient who has hep C chronic infection, but no signs of liver damage. Such an individual might choose today to postpone treatment. This is not a recommendation, I'm just constructing a scenario, but it is the case that sometimes such an individual will choose in consultation with their doctor to postpone treatment until newer therapies are developed because there isn't yet liver damage. For those individuals, if they knew that they had the good response genotype and had a better than 80% chance of being cured by the current treatment, they might decide to go forward. So one can construct scenarios where the information would be relevant to the decisions that are taken. And for those sorts of reasons, all of us involved in the study do feel that this information should be made available to both clinicians and patients as they are considering options. Now, the way that would happen is relatively straightforward. The intellectual property associated with the discovery is owned and controlled by the drug company, Schering-Plough, and they would have a couple of options in how they would make it available, either selling the rights to the test to one or more companies that would then provide it as a diagnostic, or doing it themselves. My expectation is that that will be done relatively quickly, because I do suspect that there will be interest in individuals knowing, as I described, their status at this gene.
Very good. Viewers could join this Duke Office Hours conversation in 3 ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. Now, Professor Goldstein, there's some even bigger issues to get at here, but before moving from specifically this study, you were hinting at some of its limits. It's obviously a very significant finding, but there's limits in terms of determining actual causation and being able to guarantee or not guarantee successful treatment for hepatitis C. So what are the boundaries you'd put around these findings?
Well, I think in terms of predicting who's going to respond, we have explained some, and in fact even a lot, of the variability amongst patients, but most certainly not all of it. So we still can't say for certain, even individuals with a good response genotype, we can't say for certain whether they'll be cured. And those, of course, with the poor response, some of them will certainly be cured by the current treatment. So the prediction is certainly far from perfect. The other perhaps important point to make is that we don't understand why this genetic difference has its effect. And that's important to work out, because if we can understand why individuals with the good response genotype respond so much better, then we might be able to develop add-on therapies that would help those with the poor response genotype to respond more like those with a good response genotype. So for example, we think that it's possible, though we don't know for sure yet, we think it's possible that the difference is due to a genetic change that results in the substitution of one amino acid in the protein that this gene makes. And then having one sequence for that protein is better than having the other. And if we're able to show that that's the case, then it's possible that the protein with the favorable sequence would in fact be helpful as an add-on to therapy. So one of the important directions that we're trying to move in is to try to develop a detailed understanding of the biological basis of this association that's been observed.
Very good. Now, you mentioned in summarizing the study that race and ethnicity was already a factor in treatments for hepatitis C. There was a documented difference by ethnicity, and we've already got a couple questions lined up in this area, but could you just lay out for us what your study, the twist that your study put on this racial aspect to hepatitis C treatment?
Well, as I mentioned earlier, it's been known for a long time that there is a difference in response amongst different racial and ethnic groups. And in particular, individuals of European ancestry have about a 50% chance of being cured by the current standard of care treatment, whereas African Americans have about a 25% chance. So this is one of those areas where there is a very large difference amongst groups that's very well accepted. Now, there's been a lot of talk over the years about differences amongst racial and ethnic groups and responses to different medicines, and a lot of that, when you closely inspect it, tends to kind of fade away because it can be rather sensational to make claims about these sorts of differences, and the evidence is not always there. In this case, it certainly is the case that there are those differences, very well established, and it's a big, important difference. And when we carried out the study, as I said, one of the things that we were thinking about was the possibility it could be explained by a gene variant that was different in its representation between the groups. And what we found is that the variant in this, in and near this gene IL-28B, the variants there, the form that leads to better response to treatment is the most common form in individuals of European ancestry, whereas in individuals of African ancestry, it is not the most common form. The most common form is the form that leads to less good response. Because of that difference in how common the different forms of the gene is in the different groups, because of that difference, the variant itself, in fact, we estimate explains more than half of the difference in response between individuals of European ancestry and African Americans. And the other really important point to emphasize is that as far as we can tell, the effect of the variant is exactly the same in the different groups. So an individual with a good response genotype who is African American is helped by that good response genotype just as much as an individual of European ancestry that has the good response genotype is helped by that genotype. So even though there still is this difference, the variant acts in a similar way, and it explains much of the difference between the groups.
Very good.
The final point to make in that context is that even though we have overall this important difference in average response rates between individuals of European ancestry and individuals that have substantial African ancestry, an individual with substantial African ancestry that has the good response genotype is in fact more likely to be cured by interferon alpha treatment than an individual of European ancestry with the poor response genotype. And so what that's telling you is that the underlying relevant genotype is the important thing to look at as opposed to race or ethnicity.
Very good. Viewers can join this Duke Office Hours conversation in 3 ways. by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. We've got a question via Twitter here from J.R. Minkle. Professor Goldstein, he asked, what is a geneticist's duty when many are inclined to attach biological meaning to race?
So it's a good question. I think that a geneticist has a couple of duties, in fact, when talking about this sort of intersection between genetics, race, and ethnicity. I think one duty is to describe the work straight. I think that it doesn't help to avoid certain topics because they are sensitive, or to put any kind of a slant on what the data actually are and suggest. So I think the first thing is you tell the story straight. The story is the story, and you don't try to put any slant or spin on it. That said, I think that geneticists have to be extremely careful not to let descriptions of their work get carried away with enthusiasms about the level of genetic determinism involved in different traits. I think there is a certain tendency for the impact of genetics to be overstated, and I think that that's something in particular when one is thinking about genetics, race, and ethnicity to be very careful about, not to allow genetic results to be overstated and to leave the impression that, for example, a lot of the differences that are described amongst racial and ethnic groups do in fact have a genetic basis. Some of them do, some of them don't. I think that there is actually a tendency in the part of some of the media to gravitate towards a genetic explanation for a lot of things, and I think that's another responsibility of geneticists to make sure that that tendency is watched for and controlled in so much as it's possible.
Great. Now we've got another question along the same lines. This one comes by email from David, and he asks about, what about well-meaning politicians who don't really understand genetics? Is there, again, this explaining it particularly to policymakers?
I think that that question actually does highlight what is, in fact, a real concern. I think that there is so much interest in genetics as an explanation for some of the things that we see varying amongst people, that there really is a strong pressure to overstate. And there was, in fact, we work in HIV as well as hepatitis C, and there was, in fact, an example of this where there was a genetic finding reported that suggested that a genetic difference that is almost completely different between individuals of African and European ancestry, where you have one form that's the common form in many parts of Africa, and another that's the common form outside of Africa. There was a study that suggested that this particular genetic difference had a gene called the Duffy locus, that this particular genetic difference influenced susceptibility to infection with HIV. And the study, in fact, was not a strong study. It did not have good controls. Yet, because it suggested that the form that was common in Africa made people more vulnerable to infection, because it was suggested that made people more vulnerable. In fact, Bill Clinton picked up on it, and when he visited Africa, he said, well, we now have this genetic explanation for why there's so much HIV in Africa. And that statement was wrong from every single perspective. The initial report of that finding, other groups tried to replicate and could not replicate. We do not believe it's true in the first place. And even if it were true, it is certainly not the case that you could infer from that the epidemic of HIV in Africa is in fact due to the underlying genetics as opposed to other things like behavior. So I think there we had a real example where the genetics was dramatically overstated by a very influential individual, and I don't fault him for that. I actually fault the scientific community for not being more careful about what it states as real, because what the scientific community states has an influence on people's thinking, even when it isn't true. And so when this was then corrected, as science does, correct itself over time, I don't imagine Bill Clinton has yet said, oh, by the way, what I said earlier isn't correct. And in fact, the New York Times covered this, and I don't think they've reported anything saying, well, now 4 papers have shown that they can't replicate those results. So I think that's something that we need to be real careful of. And I suppose I would say that influential people, politicians and others, could probably do a little bit better job of being careful. For example, maybe you pick up the phone to a few geneticists before you decide to make a statement like that and say, hey, is this convincing? Is this solid? Because you wouldn't, in that case, have had to ask too many geneticists before you would have got some warning signs.
Very good. Viewers can join this Duke Office Hours conversation in 3 ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. Professor Goldstein, we've been talking here about the intersection of genetics and race, and also your research touches on medicine, how these science is applied in the clinic. The term here, pharmacogenetics. I think this may sound exciting and promising to some. For other people, it may be a little scary. So when how would you unpack pharmacogenetics for us?
So the science of pharmacogenetics has as its aim developing genetic diagnostics or predictors of response to treatment. And the hope there would be that you could tailor the treatment to an individual's genetic makeup. And of course, the hepatitis C IL-28B study allows, to a degree, exactly that, in the sense that we now know for whom the treatment is more likely to succeed. There has been, over the years, waves of both enthusiasm and despair about pharmacogenetics. I think that for a short time, many people felt that once we started looking at the genetic basis of drug response, it would usher in a transformation of medicine where we were personalizing medicines all the time by genetic makeup. Now, that most certainly has not turned out to be the case so far. It has been very much slower going than many people were hoping, and than many people were certainly, I would say, expecting as well. Whether that's going to start to change in the coming years, that we will make more progress, find more associations between genetic makeup and drug response, it's really difficult to say. Overall, progress has just been pretty slow, and that might be because our tools for looking at the genetic makeup of patients have not been mature enough. Those tools are now maturing at a phenomenal rate. Or it might be that the underlying genetic basis of these treatment responses is often just pretty complicated, and so it's hard to find cases where a single genetic difference, like we found here, has a big impact. We just don't know which way it will go.
Okay. Viewers can join this Duke Office Hours conversation in 3 ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. We've got a question here from Britt by email, and Britt asks if the underlying genotype is more important than race or ethnicity. I am wondering what the efficacy is of designing studies around racial and ethnic categories. Professor Goldstein.
So actually that's a very good question. To a certain degree, you could argue that if gene variants have consistent effects in whatever group they happen to be found in, and I do tend to believe that that's usually how it's going to work. That's my hypothesis in any event, and that is what we see for IL-28B. If that's what you think, then you could argue that it's less important to include the widest possible variety of racial and ethnic groups in studies, because anything that you find will be applicable across the groups. And that is true to a degree. The complication, however, is that sometimes gene variants have very different representations in the different groups. And therefore, if you did a study, say, only in individuals of European ancestry, there might be a variant that's important, but it's too rare in that particular grouping to see its effect. But then if you had another group of individuals, it might be common enough to see its effect. And so for that reason, even if we accept that variants have consistent effects, because of that possibility, there still is, I think, a strong and important motivation for carrying out studies in ethnically and racially diverse population groups.
Very good. So following on that, this gets to the issue of methodology and Genomewide Association studies. There was a New York Times article profiling you last year where you were critical of these studies, although in this most recent study you did, it was a Genomewide Association study. So talk to us about that.
I have, despite some of the perceptions, I have absolutely no gripes about Genomewide Association studies. In fact, I have been doing them for some time, and I was a tremendous enthusiast of the development of the tools to allow that work. My concern really is more about viewing the outcome of Genomewide Association studies as important when they're not. So my take is that, by and large, most of the traits that have been studied with Genomewide Association studies have not been very well explained with those studies. What's generally happened is that we have found a few places in the genome where there are very, very weak, but real, but weak associations between genetic differences and the trait. For example, type 2 diabetes or schizophrenia and so on. Very, very weak associations. So first of all, those weak associations are not of value in terms of prediction. So they're too weak to really say anything useful based on genetic makeup about who's likely to develop the disease or what you could do to prevent it. But more importantly, in general, with some exceptions, we haven't been able to use those associations to track down and learn more about the biology of the disease. And for those reasons, the Genomewide Association studies have not given us very much. But that's not the case for everything. For hepatitis C, for example, the Genomewide Association approach explains a lot of it and gives us a new entry into biology. I think that that's happened also in the case of looking at the control of HIV. I think it's also happened in some specific other diseases, but not very many. And the question is, why? Why have we had success for certain traits, but not so much success with Genomewide Association studies for other traits? And my guess here, and this is a guess, but my guess here is that the answer is the obvious answer. A Genomewide Association study looks at common genetic variation principally. It is essentially blind to rare genetic differences, essentially blind to them. And so what we are doing is looking only at those variants that are okay in a way, that natural selection allowed to increase in frequency in the human population. And those variants, by and large, don't make us sick. They are common in the population precisely because they don't make us sick. And I think that's exactly the reason why the returns from Genomewide Association studies have been quite modest. And I really don't like overselling the modest results of an experiment. But the experiment was a good one to do first, because you need to rule out common variation before you move on to other studies that are harder and more expensive, looking at rarer genetic differences. And also, as I said, there are certain traits where you do find things.
Professor Goldstein, you mentioned HIV/AIDS and this working hypothesis of yours of looking for rarer variants within subpopulations. And I understand that that is a strategy you're taking in terms of trying to understand the genetic factors and possible treatments around AIDS. What's your approach there?
Well, there's really quite a remarkable transformation going on in the world of discovery genetics. Over the past, let's just say, about 2 years, the technology has matured to allow interrogation of the entire genomes of individuals that you are studying. Whereas a Genomewide Association study only looks at common variants, you can now take an individual and sequence their entire genome and look at every variant that they have, with some parts of the genome that are hard to sequence excluded. But by and large, you find everything that's there. And what that allows you to do is look not only at common variants, but also to look at rare variants. So that really is a phenomenal ability. I mean, if you think about it, what I'm saying there is that you can run a genome project, the equivalent of a human genome project, because you're taking an individual and determining their sequence entirely. And that is what the Human Genome Project principally was. And now that goes on in labs, in fact, all over the world. So we are able to sequence now a couple of genomes in a week. So that's a couple of Human Genome Projects per week. So that really is a remarkable transformation. Despite all that, it still is time-consuming and expensive. And right now, it might cost, say, up to $25,000, $30,000 to sequence an individual's genome. So because of that, we can't carry out really large studies looking at hundreds and hundreds of individuals right now because it's still too time-consuming and expensive. So the design that we're thinking about to look for these rarer genetic differences that might be important is one that we call an extreme trait sequencing design. And the idea here is to look at the population for the trait you're interested in. In our case, for example, resistance to infection by HIV. And then you take the extreme distribution of all the individuals, those individuals that you know definitely cannot be infected by HIV. That's rare, but it happens. And then you look just in those individuals and interrogate their genomes entirely with whole genome sequencing, and any rare variant that causes resistance to infection with HIV will be increased in frequency in that group, and you'll be able to see it once you've sequenced the genomes completely.
Very good. Viewers can join this Duke Office Hours conversation in 3 ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. We've got a question via Twitter from Ribogenics, and Ribogenics asks about the scenario when someone gets bad genetic news. So, for example, are there other therapies for individuals with non-responsive variant of a gene?
So specifically in the case of hepatitis C, there are new treatments that are being developed right now that are in late-stage evaluation, protease inhibitors. And we don't yet know how the IL-28B status influences response when those drugs are being used. So that is a priority to evaluate. It might be the case that the individuals with the poor response genotype do better when that treatment is used. We don't know. Certainly, there are many avenues being explored right now for treatment of hepatitis C, and the hope would be that there would be other directions that would be helpful precisely in those individuals that have the poor response genotype. My expectation would be that would in fact prove possible, and that in fact perhaps the protein encoded by the gene will be a part of that.
Very good. Viewers can join this Duke Office Hours conversation in 3 ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. Professor Goldstein, you've got a book out called Shakeups Legacy, and it has a kind of Indiana Jones feel to it. You get an email from a stranger asking questions about ancient Israel, and you dive in. So let's talk about how the book came to be, and especially the adventure of genetics.
Well, every once in a while, I mean, I don't want to over-romanticize it. I mean, of course, the reality is that that day to day, it's just a lot of little tiny bits of detail that you need to look after, and it's not like there are eureka moments sprinkled through every working week. But that said, it's true that every once in a while, doing genetics, you do feel a little bit like you're in a fascinating playground, and certainly the experience with the Jewish genetic studies that we carried out were very much like that. Just one thing after another, frankly, stunned me the way that it turned out. I guess the most surprising thing, first off, was the fact that you can get a little bit of information about what's happening in history from looking in our genomes. I mean, if you stop and think about it, that's a pretty remarkable thing. I've always really enjoyed history, and I've always thought really that that's other people's business, people that, can do archaeology and things like that. But in fact, looking in the genome tells you a little bit here and there, and that to me was striking and satisfying. And then the other thing that happened is that there were some very, very specific questions that we had, and those specific questions could be formulated to make very specific genetic predictions. And in a few of those cases, it turned out to work very, very cleanly and very simply and neatly. And that was really, really satisfying and, as I said, surprising. Maybe one of the best examples of that was this group from Southern Africa that had a tradition of Jewish ancestry. And this is a group called the Lemba. And there's this tradition of Jewish ancestry, and we thought, well, a lot of groups have a tradition, but often there isn't too much support for it. But we do have this set of genetic markers on the Y chromosome that appears to be somewhat diagnostic of connections to Jewish populations. And so we said, okay, well, we can look for it. If we happen to see that particular group of markers, then we could argue that that probably means that there really is a connection to Jewish populations. And frankly, we had very little expectation that it would turn out that way. We just carried out the study because it could be done, and it would have an answer. Then we did the study, and what did we find? This particular group of markers that forms what we called a haplotype that's common in a certain group of Jewish people, those that have a tradition of being priests or Kohanim. We found that this particular Y chromosome type was in fact very common in the Lemba, which did in fact strongly suggest that there was a Jewish connection. And when you see something like that, you feel like this is really something that we can add a little bit to what we know about the histories of people by doing genetics.
Very good. Viewers can join this Duke Office Hours conversation in 3 ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. We've got an email here from Bernard, Scottsdale, Arizona, and he asks, Dear Professor Goldstein, on a less serious note, I am a Jew whose antecedents were Levites with Ashkenazic and Sephardic origins. I was tested through the National Geographic's genographic program and found to be of the haplogroup, this is specific, J2M172, which is characteristic of a significant population of Jews. The male side of my family have always exhibited facile abilities in art and calligraphy. The ancient Levites acted as scribes according to the Bible. Can this be supported by present genetic knowledge? A lot there, but it's right in line with what we're talking about in your book, Professor Goldstein.
I think the short answer I would say to that is no. The longer answer is the genetic differences that we are using to determine the relationship amongst different Y chromosomes or mitochondrial DNA or indeed other parts of the genome, they themselves are most frequently not functional so far as we know. They themselves don't have any particular consequence at the level of the individual. They're just ways of distinguishing stretches of DNA. On top of that, if you have a particular part of your genome, let's just say a Y chromosome, but it could be another part of the genome, that comes from a particular ancestor some ways back. That actually tells you very, very little about what's going on in other parts of your genome. So for example, my Y chromosome might come from some particular ancestor some number of generations in the past, but that particular ancestor may have in fact given absolutely nothing else genetically to me. So the connections that you have for one part of the genome back to an ancestor in the past are quite different from another, and so I actually see essentially zero prospect for genetically influenced characteristics being transmitted intact over multiple generations. So I don't see things working that way at all.
Very good. When we're talking about history, are we also talking about evolution? Does that play into the theory that underpins your research?
I think that one always has to be considering the evolutionary history of humans when one is doing this kind of work. And the best really example that I can give for that right now is what I was describing earlier for what I perceive as the limited returns from Genomewide Association studies. I think that the explanation there really is, as I said, related to natural selection, and it's sensible for us to be thinking about the selection pressures operating in humans as we are designing our experiments and thinking about what to do next. I also think that it is likely, though this is absolutely not proven, but I think that it's likely that selection may be responsible for what we saw in the hepatitis C study. The variant that leads to better response to treatment, as I said, is very different in its representation in individuals with ancestry from different parts of the world. As I said, it's more common in individuals of European ancestry than African ancestry, but it's yet even more common in individuals from East Asia, the good response form. And I think that it is quite likely, in fact, though I said not proven, that the reason that it's distributed as it is geographically is that the form that we now see as a good response form in response to treatment actually helped defend against some virus or set of viruses that different human populations experienced to different degrees in history, and there was therefore selection that increased the representation of what we now call the good response form at IL-28B.
Very good. Viewers can join this Duke Office Hours conversation in 3 ways: by emailing live@duke.edu, visiting the Duke University Facebook page, or using the Twitter tag Duke Live. Professor Goldstein, we were just talking about recent human evolution when you're considering your genetic research. Is it right to apply the word social Darwinism, or is that talk to us about that recent human evolution?
Social Darwinism is not a term that I would use because it means too many different things to too many different people. I think that the real point is that there's no doubt that the structure of, and I'm only talking about genetic variation here. I'm talking about the selective forces that influence genetic variation. There are, of course, many other questions about the role of selection in the development of humans that don't relate to genetic variation, but what I study is genetic differences amongst people, and I'm talking about those. I think that it's quite important that we do consider how the genetic variation that we carry as a species has been shaped by our evolutionary past. I wouldn't take it all that much farther than that myself. I wouldn't take it to generating theories about our behavior on the basis of evolutionary models. That's not the direction that I prefer to go. It's much more just thinking in terms of, well, if a variant has an impact on a trait, either predisposition to disease, resistance to infection, and so on, then selection will influence how much of that variant we see where we see it, what other variants in the genome might be interacting with, and so on.
Very good. We've got one pretty scientific, technical question here for you, Professor Goldstein. It comes from Furneaux by email, and Furneaux asks, if a common variant regulates gene expression, is it possible that they indeed cause disease? And then the note here, since their disease phenotype may depend on environmental signals that regulate transacting factors that in turn regulate the variant.
So that's a good question. One of probably the leading hypothesis right now for why we are seeing some associations between common variants and diseases. As I said, they are not very strong effects, but they are there. Probably the leading hypothesis is that what's happening is that there are genetic variants that have a small impact on the expression of a gene, and then that small impact on expression ends up creating a small impact on risk of disease. And I think some of that must surely be happening, that some of the genetic variation that's related to disease does have its effect through small effects on gene expression, and then that ends up creating a predisposition to disease. And the point has been made, which is a fair and accurate point, is that even if the effect of the genetic variant on disease is itself very modest, it could be that it gives you a pointer to the gene, and that if you pharmacologically acted on that gene more strongly than the genetic variant does, you might be able to produce a strong effect in terms of controlling the disease or reducing risk of disease. And that's true. So I think that's probably happening. How much it's happening, we don't know. And the reason we don't know is that in the vast majority of cases where these associations have been made between common variants and disease, we've never tracked down to the cause. So time will tell whether it turns out that way. The other thing to mention in the question about gene expression is that although the study of common variation turned up relatively little for neuropsychiatric disease, on the other hand, the just really burgeoning now studies of rarer variants have shown that very large deletions in our genome are clearly very strong, important risk factors for some neuropsychiatric diseases. In particular, it's been shown for schizophrenia, autism, and epilepsy. And here what happens is we have for most of our genomes, we have 2 copies of genes, one we get from our mother and one we get from our father. And what happens is that in some parts of the genome, you can have a deletion that removes several of those genes from one of the copies that you have. And so instead of having 2 copies of say, 6 genes in a row, you have only 1 copy. And that reduces the expression of all the genes that are included in that deletion. And we now know that those deletions can have very strong impact on predisposition to disease. That's probably happening through expression, although it's not certain.
Very good. Professor Goldstein, you've been very generous with your time in holding these office hours. To close out, we likely have some students watching, present here via internet. And I wonder if you would conclude by just a message to young people considering the field of genetics.
Well, I suppose what I'd say is that hurry and join the party, because there really just couldn't be a more exciting time to be doing genetic research. What we are doing right now can be done for the first time only once, and what we are doing right now is characterizing what human genetic variation is like, and we're doing it comprehensively and trying to relate that to differences amongst people. And the work will be going on, to be sure, for a long time, but that first look is happening in the coming years, where for the first time we can actually take large groups of people and find all the genetic differences that are there and ask what they do. That's the work that's going on right now, and it really is a thrilling time to be doing genetics.
The conversation continues online. Email live@duke.edu. visit the Duke University Facebook page or use the Twitter tag Duke Live. To learn more about Duke, visit duke.edu.